네, 저는 현재 NYU 정신과 프로그램에 있는 전희철입니다. 제가 사실 최대한 한국말로 발표를 하고 싶었는데, 한국말도 좀 서투르고 영어도 서투른데, 그나마 영어가 좀 더 편해가지고 영어로 발표를 하고, 어, 그 Q&A 세션 때는 최대한 한국말로 답변을 드리겠습니다. I just wanted to briefly introduce myself before proceeding to my research work that was recently published. I was born in Korea, immigrated to Canada, and then I came down to college for United States. I went to Johns Hopkins University and I met Dr. Go Li Ming and Dr. Hong Jun Song, where I got engaged in the basic science research for the first time. And here I was working in the topic relating to adult hippocampal neurogenesis. And from there, I was working with a postdoc at the time. Her name is Dr. Mihan Jang. She, at the time, got a faculty offer from Mayo Clinic in Rochester, Minnesota. So basically, I followed her to work with her and also to help her set up the lab. And from there, I was applying to MD PhD programs, and I got accepted at University of California, Irvine. And I went there for my training and met my PhD mentor, Kei Igarashi. And with him, I've been exploring learning and memory in the context of entorhinal hippocampal circuit, both in the disease state as well as the healthy brain state using the rodent models. And just recently, I moved to New York City and I joined NYU as a research track psychiatry resident. And currently, I'm spending half my time in the clinic and also half my time in the research training as a postdoc at Yuri Buzaki's lab. Like I mentioned, I'm currently at NYU, and my role is involved in both clinic and the research training. So I'm trying to really do my best to kind of juggle between the both. Just before jumping into my research, the publication, I wanted to give a preface and also introduction to kind of the area of the research that I've been exposed to. So this is just a background. So when it comes to entorhinal and hippocampal circuit, there's been extensive studies done relating to understanding the spatially relevant neurons involving place cells found in the hippocampus, mostly in the C1, and also the gray cells from the medial entorhinal cortex. And together, they're thought to be very important neuronal correlates for spatial navigation and memory. And they've been discovered by the Mosers and John O'Keefe, and they won the Nobel Prize in 2014 for these discoveries. Now, the gray cells are found in the medial portion of the entorhinal cortex, and there's thought to be a lot of spatially relevant neurons involving border cells, speed cells, and head direction cells. However, when it comes to the lateral portion of the entorhinal cortex, there still remains a lot of mystery. So, what we know for a fact is that the lateral portion of the entorhinal cortex receives a lot of olfactory input, at least in the rodent models. So we wanted to really explore this area in the context of kind of olfactory related learning tasks. So just to give a little more background on the lateral entorhinal cortex, previously there's been a lot of studies on this, mostly done in the rats that are very significant. So there's been some egocentric coding cells that were discovered by the Jim Canarian's group back in 2018. And also Albert Zhao showed that there's kind of a temporally um, temporal coating in the lateral portion of the entorhinal cortex, also found in the rats. And also Takehara from University of Toronto demonstrated that there's kind of this state dependent or the situation cells found in the lateral portion of the entorhinal cortex. So with all these differing coding and findings in the lateral portion, how can we really reconcile the role of the LAC? And really to address that, we use the rodent mouse model, the genetic model, and we really combined EFIS with optogenetics to study specific cell types within the LEC. And also, we wanted to perform the optogenetic experiment to assess their functionality by performing the causality experiment. So this is the behavior test that we have designed to really investigate the role of the LEC. Because the LEC gets a lot of input from the olfactory region, we figured that our learning task should be in the context of the odor cues. And therefore, basically, it was a go, no go task that we have designed where we have taught the animals to associate a particular order A in this case, as a reward, and order B as a punishment. So basically, the animals learn to lick in response to order A as they were being rewarded with a sweet or the sucrose water. And they learn to not to lick in response to order B, where they were presented with kind of a bitter or quinine water that would be very aversive to the rodents when they were licking. So about 5 to 10 days, the animals learn to kind of distinguish between go order A and no go order B. And once they became familiar, we started introducing them with a novel pair of order where they had to figure out whether that order was going to be rewarded or whether that order was going to be punished. So on day one, they'll be presented with order C, which would be novel go order, and order D, which should be considered as the novel no-go order. And on consecutive days, they were presented with more novel pairs of orders, E, F, G, H, and so forth. So basically, this is the learning performance, the performance curve for the animals. If you look at the blue tracing is showing the performance curve for the familiar order A and B, where the animals are already acquainted. And the red tracing is demonstrating the performance curve for the novel pairable order, where the animal had to distinguish and acquire the new information. So this task was really designed to distinguish between the memory retrieval and the acquisition. The retrieval in this case would refer to order AB, which are the familiar orders, and the acquisition would be referring to the novel pair order as shown in the red tracing. And really, our idea was that the lateral antennal cortex is important for acquiring the new information into the memory during this associative learning task. So this is kind of the video to illustrate the task of the behavior. So here the animal's presented with a go order, animal has to wait, and when the LED, the instructions given, they have to learn to lick. And if they lick, they'll be rewarded with sucrose water. And here they're presented with no go order, and they basically have to learn to not to lick when the instruction LED is given. So this would be considered as a correct trial. Now the last trial is showing no-go trial where the animal actually accidentally licked, and that will be considered as an error trial. So I'm kind of also giving background to our previous work, but here We basically demonstrated that the superficial layer of the entorhinal cortical cells are very important for integrating the rewarded information. And really, the rationale behind this project was that um, it's actually the superficial layer 2 and 3 that receives the majority of the olfactory input. So we figured that if we block the area where it's receiving the olfactory input, that's going to impair the animal's ability to acquire new information. And also the superficial layer 2 and 3 are the areas that actually send projections to the hippocampus. So we thought that this would be more pertinent to learning and memory function. So there's fan cells in the layer 2A and pyramidal cells in the layer 2B/3. So we first try to assess their function by optogenetically manipulating them. And what we saw was that there was basically impairment of the acquisition of the novel odor when we were inhibiting the fan cells, but not the pyramidal cells. And from this, we wanted to record what these fan cells were coding for. And when we recorded them, basically, they demonstrated that the fan cells were coding for the novel rewarded information. As you can see, the population activity shown on PC trajectories at the bottom. You can appreciate that the novel order, one is slowly integrating with the familiar order A as the animals learn the task, whereas the novel order 2 and order B kind of remain at a separate domain um, even after learning. So, what's the mechanism behind this? Basically, our high suspicion was that there will be a kind of a reward signal feeding information into the lateral entorhinal cortex. And what we basically demonstrated was that the fan cells in the LEC require dopaminergic input to really integrate the novel rewarded information for this associative learning. So basically, that's what we showed, and we published this a couple of years ago with my colleague, dear colleague, Jason Lee, who is now a postdoc at Allen Brain Institute. And it was really, really fun work. But at the time, one of the main criticism we got from our reviewers was that if the lateral entorhinal cortex is truly important for this associative or kind of the assimilative learning, how come there's no representation of both reward and punishment? Why are fan cells only coding for reward? So to really address this question and this criticism, we further probed into the other areas in the lateral entorhinal cortex. So we basically followed the circuit, went down the stream, and basically hippocampus and entorhinal cortex kind of forms a loop. And from layer 2A, the fan cells, they send projections to the dentate gyrus and the CA3 of the hippocampus, and they send projections to C1, and from C1 and subiculum back to the deep layer of the lateral entorhinal cortex. Um, so we thought that the deep layer would be next target to really focus on because it's the area that basically gets the input from it basically gets the input from the information that's processed from the hippocampus. And it's also actually the area that's highly connected to the other cortical regions. Um, so we used one of the animal models that expresses at the deep layer. Um, so the animal model that we used was the RBP4 Cree line. And with this line, basically we started with our causality experiment where we optogenetically manipulated them during the associative learning task and basically demonstrated that the deep layer of the LEC is also important for the acquisition of the novel order, whereas the pre-learned or the memory retrieval remained intact. So the next obvious question was, what kind of information is being coded by these deep layer neurons? And very surprisingly, when we optogenetically isolated their activities and recorded them, we saw representation of both reward as well as punished information that were integrating to the familiar orders as the animals learned. So this really told us that it's actually the deep layer of the LEC that represents the both reward and punished information. And when we looked at their population activity, we saw significant integration toward the reward as well as the punished order throughout the learning. Um, and basically, this is just the videos to illustrate how they appear before learning. So you can appreciate the deep layer LEC neurons are basically separated all in different domains as a whole population activity. However, once the animals learn the task, you can appreciate that the novel order, the rewarded and the punished order start integrating with the familiar rewarded and familiar punished orders. So from this, we really wanted to ask ourselves, how is this being, what other brain regions is this area being connected to? How is this information being shared and where is it being shared? So to address that, we started with a simple anterograde tracing experiment where we injected the FlexTD tomato in the LEC of the RBP4 Cree animals. And lo and behold, we were really surprised to find out that there was a very massive projections toward the medial prefrontal cortical region, particularly more so on the prelimbic and the infralimbic area. And when we did a retrograde tracing to see if there was any projections coming back from the prefrontal cortex to the LEC, we saw that there's a massive projections from the prefrontal cortical region to specifically at the deep layer of the lateral entorhinal cortex. So this really told us that there's this almost like a bidirectional loop between the prefrontal cortex and the deep layer of the LEC, and really wanted to kind of expand this study further by recording the prefrontal cortex during the associative learning task and what kind of information is being shared between these two regions. So we first started with optogenetic experiment again. In this time, we used a senescent promoter-driven JAWS and expressed them at the medial prefrontal cortical regions, and we optogenetically inhibited them during the associative learning task. And slightly different from prior inhibition experiment, we could appreciate that basically it affected both the memory retrieval as well as the memory acquisition. And when we recorded the cell activities from this area, similar to what we saw from the deep layer of the LEC, we were able to appreciate that there was a representation of the both reward and punishment that were integrating toward the familiar orders. So when we looked at the population activity, I will definitely go through this in more details later on, but we saw something very similar to what we saw in the deep layer of the LEC, except that initially, just before the animals learned, there was some more overlap between the novel go and novel punished orders. However, as the animals learned the task, they basically were assigned to either of these familiar orders. And these are the videos again to illustrate the population activity. So you can see that there's some overlap between the novel orders in the beginning. However, as the animals learned the task, these novel orders started integrating toward the familiar orders, positive and negative familiar orders. So when we basically got these electrophysiology findings, our next big question was to really find out which is kind of the chicken and the egg. Is it the prefrontal cortex that's driving the learning and the input to the LEC, or is it the other way around? So that was really hard to address by just recording from one site. And therefore, to really address this, what we decided to do was record from one region while inhibiting the other region to look at the population dynamic that evolved during the learning. So to address that, we first recorded from the prefrontal cortex while inhibiting the deep layer of the LEC. And basically, what we saw was that the familiar order A and B still remain separated, but the novel order 1 and 2 remained at the same domain, almost like there's these three attractor states. So attractor state for the punished familiar order and attractor state for the rewarded familiar order and attractor state for kind of unassigned order for 1 and 2. Just this is the video to kind of better illustrate how it appeared on the PC trajectories. And next, we wanted to do the same experiment, except looking at the deep layer of the LEC while inhibiting the input from the medial prefrontal cortex. So when we looked at the deep layer LEC cells as a whole population activity, what we could appreciate was that basically all four orders remained at different domain. And this is different from the prefrontal cortex, where the novel orders were unassigned at the same domain, kind of the same attractor state, whereas in the lateral entorhinal cortex, the novel order pairs were remaining separate and at different attractor states, basically. And just this is another video to illustrate better illustration of how these PC trajectories look. So this kind of the last slide, but I'm kind of getting straight to the point. But from what we've seen in the LEC and the medial prefrontal cortex, what we can appreciate is that they have basically a differing initial representations of the order, but eventually through learning, basically these novel pairs start integrating toward the familiar orders, kind of what I would say would be like a schema. Now, what's interesting to note is that in the LEC, they basically remain in separate domains if they're not getting input from the prefrontal cortex. So what I'm seeing this is basically the LEC is coding for the sensory features of the order that's being presented to the animal as the animals are learning, but it really requires a prefrontal cortex to help assign these sensory information into the rules. So what I want to say is that the LEC and prefrontal cortex are equally important and they're codependent on this test-dependent learning. And also something that I want to highlight is that the prefrontal cortex, on the other hand, really basically, I think is more representing as a kind of a test rule where either the one information is being presented as kind of a positive or the negative valence information, but without the identity of the information being presented by the LEC, prefrontal cortex won't be able to basically assign them into either positive or negative valence information. So what we really want to highlight from this work is that I know there's a lot of focus on the spatial coding and information in the entorhinal cortex, but we also want to really highlight that the lateral entorhinal cortex um, may code something beyond non-spatially relevant information. And in this case, we wanted to really draw a picture that LEC is important for producing a cognitive map of test relevant stimuli, and it's working closely with both the hippocampus and the prefrontal cortex as an interface to really relay this information during the associative learning. With that, I'm done with my presentation. So I'll take some time for questions and answers. Thank you.